The DNA is telling us. Doing the Y chromosome work, I found that there wasn't a link between Richard III and the male line relatives who are alive today.
King Richard III's legacy has always been shrouded in mystery. But what modern science has uncovered is far more shocking than the legends that followed him. When archaeologists first pulled his battle-scarred skeleton from beneath an ordinary parking lot in Leicester, the world was astonished. Yet no one was prepared for what the DNA would reveal next. Cutting-edge genetic analysis has now exposed details that upend centuries of accepted history. Discoveries so unexpected that historians, genealogists, and royal experts are scrambling to make sense of them. Was Richard III truly who the chronicles claimed he was? Did his bloodline fracture in ways that could shake our understanding of English monarchy? Or did the truth, preserved silently in his bones, finally expose a secret hidden for more than 500 years? The answers have ignited intense debate, revived old controversies, and forced scholars to confront a reality far stranger than fiction. Get ready to rethink everything you believed about royal identity, medieval power, and the dynasty Richard left behind, because the truth, revealed by DNA, will surprise you. The Lost King's Bones. For more than 500 years, the final resting place of Richard III was uncertain. Chronicles told of his death at the Battle of Bosworth in 1485, the last major clash of the Wars of the Roses. Accounts said his body was taken to Leicester and buried without ceremony at the Grey Friars Friary. Yet, over centuries, the friary was dissolved, demolished, and the memory of its exact location faded. By the modern era, the king's grave was thought to be lost forever, perhaps even thrown into a river, as later rumors suggested. That changed in 2012 when archaeologists launched an excavation in Leicester. The unlikely site was a council car park. Research had shown that the friary once stood in that very area, its outlines hidden beneath asphalt and concrete. Few expected that anything would be found, let alone the bones of a king. But within days of digging, a human skeleton was uncovered in the area believed to be the friary's choir, precisely where some records suggested Richard III was buried. The skeleton, referred to at first simply as Skeleton 1, carried features that instantly drew attention. It was the remains of a man in his early 30s. Richard III had died at 32. His bones bore the scars of battle, wounds to the skull consistent with blows from swords or halberds, and cuts that suggested his helmet had been removed in the final moments of combat. These matched contemporary accounts of his violent death on the battlefield. Perhaps most telling was the spine. The skeleton displayed severe scoliosis, a curvature that would have caused one shoulder to sit higher than the other. Chroniclers from Richard III's time described him as having uneven shoulders, while later portrayals, especially Shakespeare's, exaggerated this into a grotesque hunchback. The bones revealed that while Richard was not as deformed as legend suggested, he did suffer from a significant condition that would have been noticeable in life. Carbon dating placed the remains firmly in the late 15th century. Chemical testing of isotopes from the teeth showed a rich diet, heavy in meat and fish, fitting for a person of high social rank. The burial site itself strengthened the case. The Grey Friars Friary had long been thought to be Richard III's final resting place, and this skeleton lay exactly where the choir of the friary once stood. Still, archaeologists knew these findings were not enough on their own. The age, the wounds, the scoliosis, the burial place, all fit, but none were absolute proof. For history to accept that Richard III had been found, something stronger was required. That certainty, they hoped, could come from DNA. Modern genetics offered a tool that historians of earlier generations could never have dreamed of. the ability to read a genetic signature preserved in bones and compare it to living relatives. If the genetic code in the skeleton matched that of known descendants of Richard III's family, the identification would be far stronger than any physical clue. The search for relatives began. Richard III himself left no direct descendants. His only legitimate son had died in childhood. But scientists could look sideways at the family tree. His mother, Cecily Neville, had many children, including daughters whose descendants stretched into the modern world. Through careful genealogy, researchers identified living people who could be traced directly through the female line back to Richard III's sister, Anne of York. These individuals carried the same mitochondrial DNA passed unchanged from mother to child. The team also explored the paternal line using the Y chromosome passed down from father to son. Since Richard III had no sons, the scientists turned to descendants of the broader Plantagenet family, particularly from Edward III, Richard's ancestor. If the Y chromosome of the skeleton matched those of the descendants, it would confirm that the remains belonged to a true Plantagenet king. The stage was set. Archaeology had delivered a compelling candidate for Richard III's remains. Now genetics would be asked to settle the question. The science behind a king. When the skeleton was finally uncovered, everyone immediately turned to science for answers. Archaeology had built a convincing argument, but people wanted absolute certainty. Could DNA taken from bones buried for more than 500 years really prove that this was Richard III? The challenge was huge. Ancient DNA is delicate, easily contaminated, and often so damaged that it becomes nearly unreadable. But if scientists could make it work, it would offer proof stronger than any written historical record. The first step was figuring out which types of DNA would be useful. Most of our genetic material is mixed together every generation, half from the mother and half from the father. But two special kinds of DNA pass down through family lines almost unchanged: mitochondrial DNA and the Y chromosome. These became the key scientific tools. Mitochondrial DNA, or mtDNA, is passed from a mother to all of her children, completely untouched by the father's genetics. This means siblings who share the same mother also share the same mtDNA, and so do all descendants along the female line. For Richard III, researchers traced this maternal line through his sister, Anne of York. After centuries of marriages, movements, and branching family trees, genealogists tracked down two living people who could be connected to Anne through an unbroken line of mothers. Their names were Michael, a furniture maker in London, and Wendy, a genealogist living in Canada. If the mtDNA from the skeleton matched theirs, it would be powerful evidence that the bones really belonged to Richard III. The paternal line, however, presented another kind of challenge. The Y chromosome travels from father to son, staying mostly the same with each generation. In theory, Richard III's Y chromosome should match that of men alive today who descend directly from the Plantagenet kings. Looking back through history, scientists focused on Edward III, a distant ancestor. From him, several male lines survived into modern times, especially the Somerset family, descendants of Henry Somerset, the 5th Duke of Beaufort. If their Y chromosomes matched the one from the skeleton, it would add another strong layer of proof. But collecting usable DNA from a centuries-old skeleton was extremely delicate work. The remains were handled only in controlled labs, where even a single stray hair or tiny skin cell could ruin everything. To avoid contamination, scientists worked inside sealed rooms, wearing full protective suits. They drilled into the teeth and into dense parts of the bones, areas most likely to have preserved DNA despite the long passage of time. From these samples, Small pieces of genetic code were extracted, carefully cleaned, and then amplified, copied millions of times until they could be read. It was incredibly precise work. Ancient DNA often breaks down into very short fragments, sometimes just a few dozen base pairs long. Sequencing machines then had to piece these fragments back together, like trying to rebuild a shredded book. Even after that, researchers had to check the results again and again to make sure the DNA truly came from the skeleton and not from soil, bacteria, or modern handling. When the mtDNA results arrived, the evidence was unmistakable. The sequence from the skeleton matched both Michael and Wendy. The chance of that happening randomly was extremely small. To many people, this was the strongest proof so far that the bones found beneath the car park were those of Richard III. But DNA can create new mysteries even as it solves old ones. When researchers turned to the Y chromosome results, they were surprised. The skeleton's Y chromosome did not match the men descended from the Somerset line. Instead of confirming the paternal connection, the test revealed a confusing twist. Somewhere across the many generations between Edward III and the present day, a break had occurred in the male line. In simple terms, a father listed in the historical records had not actually been the biological father. Scientists call this a false paternity event, a polite phrase for what basically means that somewhere in the royal lineage, an illegitimate child had been accepted into the family line. This could have happened in Richard III's branch, in the Somerset branch, or anywhere between them. The discovery brought up uncomfortable questions. If the skeleton's Y chromosome didn't match, what did that imply about the true continuity of the Plantagenet bloodline? The findings sparked a wave of speculation. Historians debated where the break might have occurred. Did it happen early, in the generations just after Edward III, when legitimacy was already a politically charged issue? Or did it come later, perhaps closer to Richard III's own lifetime? Without more DNA samples, the exact point of the break remained impossible to pinpoint. But one thing was certain. The royal bloodline, long seen as a flawless and carefully preserved thread through history, was not as perfectly unbroken as many had believed. For scientists, the strong mtDNA match was more than enough to confirm the skeleton as Richard III. For the public, though, the Y chromosome puzzle added a dramatic twist. It hinted at hidden secrets buried for centuries within one of England's most famous royal families. Instead of closing the story, the DNA evidence opened a new and fascinating chapter, and it raised even deeper questions. If Richard III's paternal line was uncertain, what did that mean for the claims of legitimacy made by the kings who came after him? The mystery grows deeper. When scientists revealed that Richard III's Y chromosome did not match the men believed to be his modern male line relatives, it sparked a surge of curiosity. The skeleton had already been identified with strong confidence through mitochondrial DNA, but the paternal line had revealed a troubling gap, a fracture running through the royal family tree. What exactly did it mean? And even more importantly, where in the lineage had this break appeared? Historians and geneticists began working backward through the generations. The Y chromosome comparison had involved descendants of Edward III, a king who lived more than a century before Richard III. If the skeleton's Y chromosome did not match theirs, then the break could have occurred anywhere between Edward's era and Richard's own lifetime. That left a wide range of possibilities, each with dramatically different implications. If the false paternity event had happened early in Edward III's descendants, perhaps in one of his many sons or grandsons, then the legitimacy of later Plantagenet monarchs could come into question. In that case, Richard III might have been just one of several kings whose biological link to Edward III was not what the genealogies claimed. But if the break came later, maybe in the Beaufort line that led to the Somerset family, then the issue might lie not with Richard's lineage, but with theirs. Either way, the results hinted that England's royal succession was far less straightforward than official histories suggested. The scientific phrase, false paternity event, sounds calm and objective, but in everyday language it points directly to scandal. At some moment in the past, a child had been fathered by someone other than the man recorded in the family tree. Such situations were not rare, even among the elite. Studies of modern populations show that false paternity events happen at rates of around 1 to 2% per generation. Over hundreds of years, it would actually be surprising for a long dynastic line to avoid them completely. Still, discovering one in the direct lineage of England's kings carried enormous symbolic weight. The public reacted instantly. Newspapers imagined secret royal affairs hidden in medieval castles and whispered about betrayals that might have quietly redirected history. Was this the genetic trace of a queen's unrecorded lover, or a clandestine relationship involving a noblewoman? Historians warned people not to leap to conclusions without knowing when the break had happened. There was no way to assign responsibility or to link it confidently to any specific historical moment. The discovery was electrifying and maddening at the same time. The evidence of a secret existed, but the full story remained locked away. For historians who study the Wars of the Roses, the finding added another twist of irony. The conflict that defined Richard III's reign centered on competing claims to royal legitimacy. York versus Lancaster, both branches of the Plantagenet dynasty. Each side denounced the other as less rightful. Now, centuries later, DNA showed that the entire concept of a perfectly unbroken royal bloodline might have been flawed from the start. The wars may have been fought over a lineage that, genetically speaking, was never as pure as both sides insisted. Meanwhile, the skeleton itself continued to offer new insights. Besides the scoliosis that shaped Richard III's posture, the bones bore evidence of multiple battlefield injuries. Cuts to the skull showed that he had been struck again and again after losing his helmet. Sharp wounds to the pelvis suggested humiliation after death, possibly inflicted as his body was removed from the battlefield. These discoveries matched historical descriptions of Richard dying bravely, but being mocked afterward. And the DNA mystery only made his story feel even more layered and more human, far different from the one-dimensional villain portrayed in Shakespeare's plays. At the same time, facial reconstruction based on the skull offered the public a startlingly human image of Richard III. Using CT scans, experts rebuilt his features, producing a face that resembled portraits painted in the years after his death. Combined with the DNA evidence of hair and eye color, suggesting he likely had blue eyes and light brown hair, the image challenged older stereotypes. This was not the hunched, dark villain of the stage, but a real man whose life and death had left a physical record, other clues, and conflicting evidence. The DNA analysis finally solved the mystery of King Richard III, and it's worse than we thought. Tests had provided the actual proof of what had happened. On one hand, the mitochondrial DNA tied the skeleton directly to Richard III's family, leaving little doubt that the bones were his. On the other hand, the Y chromosome mismatch raised questions that could not easily be answered. But DNA was not the only source of evidence. Other scientific techniques and historical records offered further insight, sometimes supporting the identification, sometimes complicating it. One of the first avenues of investigation was isotopic analysis. By examining the chemical makeup of Richard III's teeth and bones, scientists could build a picture of his diet and where he had lived. Teeth record what a person consumed during childhood, while bones are more reflective of the last decade of life. Results showed a shift from a more modest diet in childhood to one rich in luxury foods as an adult, consistent with Richard's rise in status. The data revealed a man who ate freshwater fish and expensive meats, drank large quantities of wine, and enjoyed imported delicacies. This aligned with the lifestyle of a noble, and later a king. Facial reconstruction added another dimension. Using 3D scans of the skull, experts recreated what Richard III likely looked like. The reconstructed face was compared to contemporary portraits, many painted within a generation of his death. The resemblance was striking. The narrow jaw, the high cheekbones, and the determined expression matched well with depictions in the National Portrait Gallery and elsewhere. Yet DNA also contributed to this portrait. Genetic markers suggested that Richard likely had blue eyes and light brown hair, features that matched early paintings, but not later ones that had darkened his appearance to fit the image of a villain. The physical evidence from the skeleton also helped to separate myth from reality. Shakespeare's portrayal of Richard III as a twisted, deformed tyrant had cemented an image of a hunched figure. The spine, however, told a subtler story. Richard did suffer from severe scoliosis, a sideways curvature that would have made his torso uneven. His right shoulder sat higher than the left, and his ribs were compressed on one side. But he was not the grotesque figure depicted on stage. The condition would have been noticeable, but with custom armor and clothing, he could still function as a capable warrior. Indeed, the wounds on his body showed he fought until the end, even after his helmet was torn away. The injuries revealed during examination painted a brutal picture of Richard III's last moments. Blows to the skull, consistent with halberds and swords, suggested that once unprotected, he was struck down by multiple attackers. Other wounds, including one to the pelvis, hinted at post-mortem humiliation, inflicted after death, as his body was carried from the battlefield. These details supported the chronicles that described his final charge, his death surrounded by enemies, and the indignities suffered afterwards. Yet even as these clues strengthened the identification, the Y chromosome mystery lingered. Scientists compared Richard III's Y chromosome to known haplogroups, the broad genetic families of male lineages. He fell into a haplogroup found more rarely in Britain. In contrast, the Somersets carried a haplogroup that is most common in Western Europe. The stark difference confirmed that the mismatch was not a fluke of testing, but a genuine break in the line. The historical implications were unsettling. If the false paternity event occurred in Richard III's direct line, then perhaps his father, Richard of York, was not who history claimed. That would mean Richard III's claim to the throne was genetically broken. On the other hand, if the event occurred later in the Somerset branch, then the problem lay with their claim, not his. The uncertainty kept historians cautious, but the very existence of a break in the male line stirred debate about legitimacy, inheritance, and what it meant to be a king. Some truths emerged slowly, hidden in bones and genes, waiting for modern eyes to uncover them. What other things about Richard III do people not know? Let us know in the comments.